Hey guys and welcome. So we're going to be going through Edexcel AS Biology B and we're going to be looking at proteins. Okay. About 18% of your body is made up of protein. Proteins form hair, skin, and nails. The enzymes needed for metabolism and digestion, and many of the hormones that control various body systems. They enable muscle fibers to contract, form antibodies that protect you from disease, help clot your blood, and transport oxygen in the form of hemoglobin. Understanding the structure of proteins helps you develop an insight into the detailed biology of cells and organisms. Like carbohydrates and lipids, proteins contain carbon, hydrogen, and oxygen. In addition, they all contain nitrogen, and many proteins also contain sulfur. Proteins are another group of macromolecules made up of many small monomer units called amino acids, joined together by condensation reactions. Okay, so they're saying that amino acid, so on, that will make a protein. Amino acids combine in long chains to produce proteins. There are about 20 different naturally occurring amino acids that can combine in different ways to form a vast range of different proteins. Amino acids. All amino acids have the same basic structure, which is represented as a general formula. There is always an amino group, NH2, and a carboxyl group, COOH, attached to a carbon atom. So they're saying this. And this is actually important, but it's the N that comes first actually, because the carbon bond is attached to the nitrogen atom, not to the hydrogen. The group known as the R group varies between amino acids. So this is what we're talking about. This is an R group. And it's always in hydrogen. So that's the structure of an amino acid. That's the formula. They might ask you to draw that in the exam. This is where sulfur and selenium are found in the structure of a few amino acids. The structure of the R group affects the way the amino acids bonds with others in the protein, depending largely on whether the R group is polar or not. Because if it's polar, then another amino acid can also be polar, and you can form hydrogen bonds. Right. Forming proteins from amino acids. Amino acids join together by a reaction between the amino group of one amino acid and the carboxyl group of another. So this is what we're talking about. This is the amino group, and this is the carboxyl group. And look, we have an elimination of water. This is a condensation reaction. They join in a condensation reaction, a molecule of water is lost. A peptide bond is formed when two amino acids join, and a dipeptide bond is the result. So that's the peptide bond. Sometimes in the exam they'll ask you to draw a dipeptide. So that's one peptide, and that's one. So it's making two, so it's dipeptide with a peptide bond. They'll ask you to circle it. The R group is not involved in this reaction. More and more amino acids join to form polypeptide chains. So we'll have another amino acid here, and the whole thing will be called a polypeptide, which contains from 100 to many thousand of amino acids. When the polypeptide folds or coils or associates with other polypeptide chains, it forms a protein. Bonds in proteins. The peptide bond between amino acids is a strong bond. Other bonds also form between the amino acids in a chain to form the 3D structure of the protein. They depend on the atoms in the R group and include hydrogen bonds, disulfide bonds, and ionic bonds. So these hold the protein together, not the polypeptide. The polypeptide is held only by peptide bonds. Hydrogen bonds. In amino acids, tiny negative charges are present on the oxygen of the carboxyl groups, and tiny positive charges are present on the hydrogen atoms of the amino acid group. Because we said, this is where, if we look at it, remember, we have delta negative and delta positive, because we said oxygen is electronegative. If we look at NH2, we said that if we look at the periodic table, we actually have this. We said the NOF. These are the most electronegative, starting with fluorine, then oxygen, then nitrogen. And so with hydrogen, which is which is weakly electronegative, so it will become electropositive as the electrons are drawn closer to nitrogen and closer to oxygen. When these charged groups are close to each other, the opposite charges attract, forming a hydrogen bond. So what happens? Should have this, but not in a crisscross type of way, but just showing that the delta negative and delta positive are attracted together. But it's not in a crisscross way, but we're just showing how they would attract to each other. Hydrogen bonds are weak, but they can potentially form between any two amino acids positioned correctly. So there are lots of them holding the protein together very firmly. Hydrogen bonds break easily and reform if pH or temperature conditions change. They are very important in the folding and coiling of the peptide chain of the polypeptide chains. Disulfide bonds. Disulfide bonds form when two cysteine molecules are close together in the structure of a polypeptide. So if we go back, you don't need to know you don't need to know the cysteine structure or anything, but it just tells you that if there were two cysteines close together, you'll have a disulfide bond between them. An oxidation reaction takes place between the two sulfur-containing groups, resulting in a strong covalent bond known as a disulfide bond. Some people refer them as disulfide bridges. These disulfide bonds are much stronger than hydrogen bonds, but they occur much less often. They are important for holding the folded polypeptide chains in place. Right. Ionic bonds. Ionic bonds can form between some of the strongly positive and negative amino acid side chains found buried deep in the protein molecules. These links are known as salt bridges. They are strong bonds, but they are not as common as other structural bonds. Okay, calling them ionic bonds is completely fine, because we know that ionic bond occurs between a metal and non-metal. So basically a non-metal would be positive because it loses the electron, and a non-metal would be negative because it would gain the electron. So that's for a metal, that's for a non-metal. Your hair is made up of the protein keratin. Some methods of styling the hair actually change the bonds within the protein molecules. Blow drying or straightening your hair breaks the hydrogen bonds and reforms them, with the hair curling in a different way temporarily until the hydrogen bonds reform in the original places. Perming breaks the disulfide bonds between the polypeptide chains and reforms them in a different place. This effect is permanent. Your hair will be styled in a particular way until it is cut. Protein structure. Proteins can be described by their primary, secondary, tertiary, and quaternary structure. The primary structure of a protein is the sequence of amino acids that make up the polypeptide chain held together by peptide bonds. So it's just the primary structure. That's it. It's a sequence of amino acids that make up the polypeptide chain, and they're held together by peptide bonds. The secondary structure of a protein is the arrangement of the polypeptide chain into regular repeating structure held together by hydrogen bonds. So we have the first type of bonds coming, hydrogen bonds. One example is the right-handed helix. So this is the alpha helix. So this is secondary structure. we have a helix. So a spiral coil with the peptide bonds forming the backbone and the R group sticking out in all directions. Another is the beta-pleated sheet, in which the polypeptide chain folds into regular pleats held together by hydrogen bonds between the amino and carboxyl ends of different amino acids. It says beta-pleated sheets. So basically, if we said that this is the primary structure, which is just a sequence, the secondary structure means it will either go into an alpha helix structure, which is spirals, or it will go into a beta-pleated sheet where they become pleated. Yeah, and both are held together by hydrogen bonds. That is their secondary structure. Most fibrous proteins have this sort of structure. Sometimes there is no regular secondary structure and the polypeptide forms a random coil. Anyway, don't focus on this bit right now. We're just focusing on this. That's the important thing to gain from this. It's just telling you something. The tertiary structure is a level of 3D. So now we're going to 3D. Or organization imposed on top of the secondary structure of many proteins. The amino acid chain, including alpha helices and beta-pleated sheets, is folded further into complicated shapes. Hydrogen bonds, disulfide bonds, and ionic bonds between amino acids hold the 3D shape in place. Okay, so here what they're telling you is tertiary structure is when it'll undergo even more folding or even more pleating, depending on the secondary structure, if it was alpha helices or if it was beta pleated. Yeah, and now we have the introduction of more hydrogen bonds and now disulfide bonds are ionic bonds. Okay, globular proteins are example of tertiary structures. Okay, so once again, I'm just telling you that globular proteins are, so they're just giving you an example. The quaternary structure of a protein is only seen in proteins consisting of several polypeptide chains. So a quaternary structure is when another now polypeptide, which is in the secondary structure or tertiary structure, joins together. So now we've got more than one type of protein of structure coming together. The quaternary structure describes the way these separate polypeptide chains fit together in three dimensions. Examples include very important enzymes and the blood pigment hemoglobin. So again, just examples. So here, the example for the secondary structure is a fibrous protein. An example for a tertiary structure was the globular proteins. An example for a quaternary structure was enzymes and hemoglobin. The bonds that hold the 3D shapes of proteins together are affected by changes in conditions such as temperature or pH. Even small changes can cause the bonds to break, resulting in the loss of the 3D shape of the protein. We say the protein has denatured. You may have come across this in GCSE. So the example would have been enzymes, and that's why when you have, so you would learn later on the temperature that kills enzymes, which is a protein, is 40 degrees. And so that's why when people have a fever of more than 40 degrees, they die, because your enzymes denature, and so the reactions that happen in your body will happen too slow to sustain life, and that's why you die. But we don't say a protein has died, we would say it has denatured. Because the 3D structure of these proteins is important to the way they work, changing conditions inside the body can cause proteins such as enzymes to stop working properly. Okay, so it's just an overview. Primary structure is the linear sequence of amino acids in a peptide. And these are just giving you examples of amino acids. Glycine being the most simplest. The R group is just a hydrogen bond, but you don't need to know that. You will get away in the exam by just saying it's a small amino acid. Secondary structure, which is the repeating pattern in the structure of a peptide chain, such as forming alpha helix or a beta-pleated sheet. So that's alpha and that's beta. Tertiary structure is a three-dimensional folding of the secondary structure. So here we can see that the 3D structure further folding resulted in even more alpha helices. So the secondary structure must have been an alpha helix structure. So Now the quaternary structure, this is a three-dimensional arrangement of more than one tertiary polypeptide. So you can see here's 1, 2, 3. So 3 have come together, and we can see the bonds here that's holding them all together. Fibrous and globular proteins. Fibrous proteins. The complex structures of large protein molecules relate closely to their functions in the body. Fibrous proteins have little or no tertiary structure. They are long, parallel polypeptide chains with occasional cross-linkages that form into fibers. They are insoluble in water and are very tough. So insoluble and tough, which makes them ideally suited to their structural functions within organisms. Fibrous proteins appear in the structure of connective tissues in tendons and the matrix of the bones, and we know that these are very strong. In the structure of muscles as the silk of spiders, webs, and silkworm cocoons. So that's in, we're talking about spiders and silkworms now. That's not the muscles. So now they're talking about different species. And as the keratin that makes up the hair, horns, and feathers. So just an example of fibrous proteins and how they're adapted for their function. So they're found, it's because they're tough and insoluble, they're going to be found in places that are also tough and insoluble. Collagen is a fibrous protein that gives strength to tendons, ligaments, bones, and skin. It is the most common structural protein found in animals. Up to 35% of the protein in your body is collagen. Collagen is extremely strong. The fibers have a tensile strength comparable to that of steel. This is due to the unusual structure of the collagen molecule. It is made up of 3 polypeptide chains, which are each up to 1,000 amino acids long. The primary structure of these chains is repeating sequences of glycine with 2 other amino acids, often proline and hydroxyproline. The 3 alpha chains are arranged in a unique triple helix. So this is what they look like. Triple helix. Arranged in held together by a very large number of hydrogen bonds. So the helixes are held together by hydrogen bonds. These collagen molecules, which can be up to several millimeters long, are often found together in fibrils that are in turn held together to form collagen fibers. They're saying that actually they're all held together. So you've got a triple helix, and that triple helix is going to be held with other triple helixes. And so the whole thing becomes a collagen fiber. So they've got a really tough structure. And collagen is what they'll pump into lips to make them bigger. It's because it's very strong, so it will stay there. Collagen fibers are found combined with the bone tissue, giving it tensile strength rather like steel rods in reinforced concrete. In the genetic disease osteogenesis imperfecta, the collagen triple helix may not form properly. The bone lacks tensile strength as a result, and it is brittle and breaks very easily. You don't have to, they're just giving an example of what happens if you're deficient in the triple structure of collagen, but you don't have to know this example. So, globular proteins. Globular proteins have complex tertiary and sometimes quaternary structures. They fold into spherical shapes. So, globular shapes just means spherical. The large size of these globular proteins molecules affects their behavior in water. Because their carboxyl and amino ends give them ionic properties, you might expect them to dissolve in water and form a solution. In fact, the molecules are so big that instead they form a colloid. Globular proteins play an important role in holding molecules in position in the cytoplasm. So basically what they said is that it will not dissolve in the water. So remember, blood has a lot of water, so if you put them in the blood, they won't dissolve. Instead, they'll be carried off to the place that's appropriate for them to carry out the action. Globular proteins are also important in your immune system. For example, antibodies are globular proteins. Globular proteins form enzymes and some hormones and are involved in maintaining the structure of the cytoplasm. Hemoglobin is one of the best known globular proteins. It is a very large molecule made up of 574 amino acids, arranged in 4 polypeptide chains, which are held together by disulfide bonds. Okay, so we've said that if this was one tertiary structure, this is another, this is another, and this is the fourth one. Those are 4 polypeptides that together make up hemoglobin. Each chain is arranged around an ion-containing heme group. So they're telling you that the very center of each polypeptide chain is a heme group. And the heme group contains iron. That's what gives hemoglobin the red color, because iron is naturally red. Hemoglobin is a conjugated protein as well as a globular protein. So conjugated means it contains another molecule within it. And here they're referring to the iron. Yes, it is the iron that enables the hemoglobin to bind and release oxygen molecules. So basically, if we don't have the iron, the hemoglobin can't accept the oxygen or release it. And it is the arrangement of the polypeptide chains that determines how easily the oxygen binds or is released. So if it was, if someone had a disease and it caused a different arrangement in that polypeptide chain, they might find their hemoglobin group might find it harder to accept oxygen molecules. So as a result, they may have symptoms of more fatigue or tiredness. So in theory, conjugated proteins. Some protein molecules are joined with or conjugated to another molecule called a prosthetic group. This structural change usually affects the performance and functions of the molecules. You have already looked at hemoglobin, a large molecule with an iron-containing prosthetic group. Chlorophyll, the molecule involved in the capture of light in photosynthesis, is another conjugated protein with a prosthetic group that contains magnesium. So you can see that the conjugated groups here are both elements. Glycoproteins are proteins with a carbohydrate prosthetic group. So basically telling you it's a protein, but they've got a carbohydrate attached onto them. And the carbohydrate here is a prosthetic group. The carbohydrate part of the molecule helps them to hold on to a lot of water and also makes it harder for protein digesting enzymes, proteases, to break them down, because if they've got carbohydrates and the protease can't bind to it. Lots of lubricants used by the human body, such as mucus and the synovial fluid in the joints. So that's the synovial fluid. If you ever cracked your fingers, you would hear pops, and that's because of the synovial fluid. Are glycoproteins. So mucus and synovial fluid are glycoproteins, whose water holding properties makes them slippery and viscous. So people with cystic fibrosis have less water in the mucus, and as a result the mucus will be drier. And so the slippery and viscousness reduces friction. This also helps to explain why the mucus produced in the stomach protects the walls from digestion. Lipoproteins. Lipoproteins are proteins conjugated with lipids. So now you've got a prosthetic group that's a lipid that's attached to a protein. And are very important in the transport of cholesterol in the blood. So you've got 2 types of cholesterol. If you learn from GCSEs, you've got HDL and LDL. And HDL is the good cholesterol. So remember HD, like HDTV, which is good. So that's going to go for your cell wall. And it builds your cell membrane. Because remember, if we looked at lipids and we said this form a bilayer. Yeah. And actually, you've got cholesterol, we'll see later on, embedded in the cell membrane. And that's the HDL. LDL, like low definition TV, is not good for you. That's what's going to be causing your plaques to your arteries to harden and causing the heart disease. The lipid part of the molecule enables it to combine with lipid cholesterol. There are 2 main forms of lipoproteins in your blood: low-density lipoproteins, which is LDLs, around 2 nanometers in diameter, and high-density lipoproteins, which are HDLs, and are around 8 to 11 nanometers in diameter. The HDLs contain more protein than LDLs, which is partly why they are denser. Proteins are more compact molecules than lipids. So proteins are more compact. So learning tip, remember that amino acids are joined together by peptide bonds to form dipeptides and then polypeptides. But the 3D structures of the proteins are the result of hydrogen bonds, disulfide bonds, and ionic bonds, but between amino acids with the polypeptide chains. So when you want to test for proteins, you would use this test. So to test for the presence of protein, you'd add 5% potassium, or you would add sodium hydroxide and copper sulfate solution, or just a burette reagent, which is the sodium and the copper sulfate mixed together. So instead of saying you would add potassium with sodium with copper sulfate, you will just say add potassium with the burette reagent, because the burette reagent is the mixture of sodium hydroxide and copper sulfate. So saying a burette reagent is totally fine. And then you add 5% potassium, you mix it together, and then you're going to take the solution that you're testing and you put it in here. If it turns purple, that is a confirmation that you've got protein present. And that's why to test for proteins is called the burette test. Burette test tests for proteins. And that's it.